Welcome to the K-State Regen Ag Pod, where we talk all things regenerative agriculture here at Kansas State University, across the state of Kansas, and beyond. I'm Nick Detter, your host, and today I'm joined by Endy Lopes Kailer, a PhD candidate from the Kansas State University Department of Agronomy with a focus in soil microbiology. Endy's PhD research as a graduate research assistant has centered on arbuscular mycorrhizal fungi, and she has received multiple awards and recognitions for her work as a graduate teaching assistant. Endy, thank you for joining me today.
Well, thank you so much, Nick, for the opportunity. I'm very excited to talk to you today and to answer all the questions that you have. So thank you for having me.
Well, I think just to start, if you could just introduce yourself, and tell us a little bit about your background and the work you're currently doing.
Yes, sounds good. So my name is Endy Kailer. Like you mentioned, I am originally from Piedade do Rio Grande, Brazil, and I've been working with Dr. Rice at K-State for the past 5 years as a graduate research and teaching assistant. I got my bachelor's and master's degree in agronomy from the Federal University of Viçosa in Brazil. And I was a visiting student at University of Kentucky 12 years ago as well. I started my PhD at K-State in 2021, and I've been working with soil microbiology. So the main focus of my research is understanding how we can use arbuscular mycorrhizal fungi, which I'll give more background soon, as a tool for improving climate change mitigation and soil health. So it's really exciting work. I am very passionate about it. I'm very lucky to have great mentors like Dr. Rice, Dr. Merberg, Dr. Blair, Dr. Dorvar, Dr. Ganga. So we had a really good group to try to find a way to see the big picture when it comes to fungal partners in soil health and how we can use them as a way to not only drive soil health, but also to um, facilitate climate change mitigation. So that's what I have been doing on the past 5 years. I also enjoy teaching. Teaching is something that is extremely important for me. So I have been teaching for every semester since I joined K-State, and I've had over 800 students at K-State, which I'm very, very happy and proud about. So the whole experience here has been incredible.
Well, I think just to start for our audience, what is arbuscular mycorrhizal fungi? And I might just call it AMF for now on, so I don't have to use that whole mouthful going forward. But what is it, and why does it matter to our to agriculture?
That is a very great question. So, AMF is a group of soil fungi that associates with around 80% of all the living plants. So, they form what we call a symbiotic relationship where they're closely together, they're closely working and living together. So, the way that this relationship works is the plants will provide the carbon that the fungus depend on. So keep in mind that the fungus cannot do photosynthesis, so they cannot produce their own carbon. And they also lack saprophytic capabilities, which means that they cannot eat the food that is around them. So they cannot feed on organic carbon. So the only way for AMF to survive is to form that symbiotic relationship with a plant. And there's some data showing that AMF was responsible for bringing plants from aquatic ecosystems to the land. So we have some fossil records that date back all the way to over 400 millions of years ago, showing that plants had mycorrhizae before they had roots. Root systems were created to, how do I say, to protect mycorrhizal structures. So essentially, it's a symbiotic relationship. So, like I mentioned, they live together, they associate with the roots of most of the plants. And in that relationship, the plant is going to feed the fungus with the carbon that they depend on. And in exchange, the fungus will expand the reach of the root systems, sometimes up to 300 times of what the roots would be able to reach on their own. So I want you to imagine like a spider web under the ground. That's how the hyphal system looks like, which is incredible. So imagine you just have an extension, a long arm that would allow you to explore areas that you'll not be able to explore, which is fantastic. And what is even more important about the fungus is that since you're exploring areas of the soil that you would not be able to reach plant-wise, they're reaching nutrients that would not be available otherwise. So a good example for that is phosphorus. we know that phosphorus is fairly immobile in soils. So unless the plants can reach that phosphorus, they're not going to uptake that phosphorus. So what AMF does is not only improve the reach of the plant to get to phosphorus pools that haven't been explored, but they are also capable of releasing organic acids that would turn unavailable forms of phosphorus into plant available forms. So that is extremely significant for plants because you can decrease the amount of external inputs that you need in your farm once you increase your population of AMF. And they tend to be pretty famous for phosphorus, also for zinc, because they don't move a lot in soils, but they are also very significant for any of the micronutrients, nitrogen, potassium, and even for water. So there are several papers showing that AMF improves water relations. So they're capable of accessing water in the soil that the plant itself would not be able to reach, would not be able to uptake, but also by forming soil aggregates, macro aggregates that will keep soil carbon and improve soil structure. And in exchange, when you improve soil structure, you're also improving water retention as well. So we have several different benefits from AMF. I could be listing them all day. I'm a big fan of AMF. For me, they are fantastic. They are one of the most important groups of soil microorganisms. And what is really interesting about AMF is that they have several layers. So if you study them to what they do, like themselves, you can talk about improving soil structure, improving nutrient use efficiency. But there's so many other studies showing that AMF also improves plant resilience to stress. Because imagine you have that network under the ground connecting neighboring plants. So imagine that in a field level. Plants can alert other plants about pests and pathogens and brace themselves and activate defense mechanisms early on when they have a strong mycorrhizal network. So there are several reports of plants being more resilient to fungal diseases, to other pathogens when they have a strong mycorrhizal connection. And you also asked like why does it matter to agriculture? I think it's a whole holistic mechanism because it connects plant health to soil health and ultimately climate health as well. Because once you're protecting that carbon inside of soil aggregates, you're keeping carbon where it truly belongs instead of in the form of CO2 in the atmosphere.
So I think I have a couple. That's first of all just amazing, very interesting. Something that I don't think about all that often. That there's these fungal networks underneath the soil and how much they're really driving the dynamics between the nutrients and, like you said, even the water. I mean, it's just kind of mind blowing. And I like that image of like the spider network. I think that's a good way to think about it. But a couple follow-up questions I was thinking as you were talking is my first question is this true for all crops? Like, is this something that happens? Is it only specific crops that we see this happen in? Because like some bacteria are like inodulation and stuff like have specific relationship with specific crops. I'm curious if this is true for all crops. And then my second question is this something that's like naturally occurring in the soil, or is it something we have to add to the soil? And if so, how do we promote and protect these AMF with our agricultural practices?
These are great questions. So the first part of your question, is it true for all crops? Not necessarily. So overall, it is estimated that around 80% of all the living plants associate with AMF. There are also all other types of mycorrhizae. So there's one type that is specific for ericoid, for like blueberries, for example. Another one called ectomycorrhizae that usually associates with trees. So we have several different layers. The most popular one is AMF that associates with around 90 to 95% of all the crops. So it tends to be the most significant one when it comes to agriculture. So that's why we put this much focus on AMF. But some of the plants like Brassicaceae family, for example, so like radish, canola is a good example too, turnip, they do not associate with AMF. And what is interesting about the Brassicaceae is that they release a compound called glucosinolates, and they are responsible for repelling AMF. So it's not like
Hmm.
the plant would lack the capability of form the association. They could, but that compound acts as a repellent. Like we have insect repellents, that would be an AMF repellent too. So that's why usually when you grow radish or you grow a non-host crops like canola or turnips, you try to avoid a highly dependent mycorrhizal crop right after it because you might lose
Interesting.
your natural populations. And you also asked, does it naturally occur? Absolutely. Yeah. So that would depend on your soils as well. So I'll give you one example from soils from Kansas. So we're in Manhattan, Kansas right now, and we have soils that used to be prairie soils. And research has shown that prairie grasses are extremely dependent on AMF. They co-evolved together over the years. And as you probably remember from the display that we have in the agronomy department, they have deep root systems. And yet they're still highly dependent on AMF for functioning, for phosphorus, and for keeping plant resilience. So soils that originated from prairie, they tend to have that natural strong population of AMF in that place. But we also know that agricultural practices, and that was part of my research as well, have been declining the amount of natural AMF. So imagine that spider web structure that I mentioned early on. It is creating an underground connection between the fungi and neighboring plants. When you till your soil, for example, you're breaking those connections. So ag practices such as conventional tillage is known for decreasing the amount of AMF progressively. And as I mentioned early on, AMF is a symbiotic relationship, and we call it a mutualistic relationship because they're both getting benefits from each other. The plants are getting more water, more nutrient, more resilience, and the fungi is getting that carbon. They also get lipids from the plants as well, but it's more into the biochemistry side of things. So I don't need to go that deep. But the point is they're helping each other. When you over fertilize your soil, you tend to repel AMF because AMF cannot
Hmm.
complete their life cycles unless the plant is willing to pay that carbon price. So, what usually happens is if you build high enough phosphorus levels in your soil, you tend to eliminate part of that AMF because they're not going to complete their circle, their life cycle, and produce more spores to the next seasons. So it's like hiring someone to clean your house after you've just cleaned it. Like for the plants, it doesn't make biological sense. So colonization rates tend to decline, and spore propagals would also decline, which is pretty interesting to look at. Um, and what was the third part of your question?
I think you actually kind of just answered it, just like in terms of ag practices, ag management, what can we do to help promote AMF? And I mean, I don't, do you have anything other to add than other than the two things you just kind of hit on?
Oh, yes, yes, absolutely. So usually it's a parallel. So with no-till, you tend to um, give them the environment that they need to thrive. So AMF populations tend to increase with no-till, and conventional tillage tends to decrease natural AMF. So like I mentioned, they tend to naturally occur, but ag practices can slow down or even decrease population. That will depend on the soils that you have too. So I've worked with some oxysols from Brazil that did not come from prairie grasses, and the amount of natural AMF was extremely low. We would get around 20% of root colonization in non-inoculated soils. And here we found up to 80, 85% without adding anything. So just natural AMF. Another thing that is really important, so like I mentioned, they need the plant to complete their life cycles. So keeping a continued live hub of live roots year round is extremely important. And that is part of my research too. So we learned that cover crops significantly increase the amount of AMF, not just in the soil, but also making that connection to the plant, which is fantastic. So if you imagine the more roots that you have, the more AMF is going to get connected to those roots. And then at the end of the season, they tend to reproduce, release spores, which are the resting structures that will then germinate and connect with other plants in the next season. So we saw in one of our research sites at the Kansas Soil Health Partnership that treatments with cover crops significantly increase the amount of AMF in the soil and connecting to the plants as well. I haven't studied crop rotation on my studies, but I've seen papers on that too, showing that rotations would also promote AMF because different root exudates would recruit different types of microbes and different types of AMF. So it's always better for us to have a more diverse population of AMF than a one species only, because if you have one species only, and that species might be sensitive to drought or to, I don't know, a specific pesticide. So if you get rid of that one, you're done. But if you have a more diverse hub of fungal partners, they will be more resilient to adverse conditions, which is extremely important. And crop rotation is a good way of achieving that. Um, so usually soils that are highly fertilized would also decrease the amount of AMF, like I mentioned. So we've seen reports of nitrogen fertilization too, but the main driver is phosphorus fertilizer. And this is one of the questions that we're hoping to answer with my research. We're still collecting data, but by the end of the year, we're going to have all of the data. We want to know and to inform farmers across the state, like how much P can I have in my soil, but still have a response from native mycorrhizae? After how much do I start losing my natural AMF populations? So this is one of the questions that we hope to answer. We want to find that sweet spot, like, hey, up to, I don't know, 35 parts per million melic-3, I'm fine. After 40, mycorrhizae declines. And this is just a random number because we're still getting data. So by the end of the year, I'll be able to provide you with a better number for that.
Yeah, I think that's a good transition to my next question. So, what have you guys been researching regarding AMF, and what have been some of the big takeaways to this point?
Yeah, that's a good question. So, throughout my whole PhD program and also on my master's program back in Brazil, I've been studying AMF, and we are trying to understand how farmers can benefit from AMF. So, like I mentioned early on, they tend to occur naturally, but with years of farming, and it's normal for many farmers to be tilling for years, and they might be transitioning to no-till now, but that was probably enough to lose a big part of their AMF populations. So there is a great interest now on learning how to bring back AMF to soils where AMF have been depleted. So we wanted to test, are bioinoculants an effective tool for bringing AMF back to our fields? So the main project that I have for my PhD is testing an AMF bioinoculant from Valent Biosciences. And that inoculant contains 4 species of AMF, so it tends to be a more diverse inoculant. And it is applied in furrow when we plant. So we wanted to test what happens when we add a bioinoculant to our field. Does that make a difference? Are we going to see any improvements by adding AMF to that soil? And we wanted to test 2 different situations. So we're testing on an irrigated field and in a dryland field. So we have a dryland field in Manhattan, Kansas, and we have an irrigated site in Scandia, Kansas. We are also testing them on soils that are naturally rich on AMF. So we have one of our fields in Konza Prairie. So we wanted to see like our soils from the prairie responding differently. And we also wanted to answer the question of how much phosphorus can a farmer have in their field, residual phosphorus, and still see a response from bioinoculant and natural AMF. So we're also manipulating levels of phosphorus. So we have 4 crescent rates of phosphorus, is starting on what would be the recommended phosphorus supply for growing corn in that specific soil with low phosphorus, going all the way to a very high rate. So around 120 pounds per acre of P2O5 is a good example of a really high one. And we've been inoculating and fertilizing every year in our dryland site. And we are doing crop rotation, so a corn soybean rotation in the irrigated site. And we always just inoculate and fertilize when we're growing corn. And what is neat about our study is that we are producing data across 5 years. And most of the studies that you see on AMF, they're either short-term greenhouse studies, or if they are field study, they're a year long field study. We want to know what are the lasting impacts of adding bioinoculants to our soils. So that way we can better inform farmers on what they would expect when they buy those products. Because unfortunately, we don't have the proper regulation that I wish we had on biologicals. So there are several products out there that might be scams. Farmers might be spending their monies and sometimes even adding pathogens to their soils or adding dead propagals, things that are not going to help at all. So we wanted to understand what happens when we add bioinoculants. And instead of just monitoring the changes on AMF, we're also monitoring changes in soil health. We want to know how the rest of the microbial community is responding to that new partner. It's like for you arriving to a party that is already full of people that you don't know. Like, how well are you going to mingle? Like, are you going to stay in there or are you going to get out of there soon? So that's what we're trying to look at. So we look at bacterial populations too. We look at actinomycetes, saprophytic fungi, the total microbial biomass as a whole. Like what are the responses when you keep adding an external partner, an external fungal partner? And more than just monitoring how microbial populations are shifting, we're also looking at processes too. So we look at enzyme production, for example. One of the enzymes that I study is acid phosphatase. So acid phosphatase is known for mining organic phosphorus from the soil. So most of our soils, they have enough phosphorus, but phosphorus is bound in forms that are not accessible. So what acid phosphatase do is dissolve that phosphorus so they can convert the organic phosphorus that is unavailable into inorganic plant available phosphorus. So we are monitoring what happens when you over fertilize, what happens when you add bioinoculants to those soils. And we collected our last soil sampling 2 months ago, and we're now processing the samples. So I believe by the end of the summer, we should have the answers that we are looking for. But just to give you just a sneak peek, like spoilers, we had years of data. So we did our first soil sampling was at baseline. So before we added any of the treatments, just for establishing the experiment. And then we did a year 2 sampling to see the effects after 2 years of experiment. And now we're going to do after 4 years of experiment on year 5. So what we saw in year 2 is that the dryland site was not showing significant responses to the inoculant on soils, but it was showing on plants. But our irrigated site was highly responsive. We saw up to a 6-fold increase in aggregate production with the inoculant,
Wow.
which was pretty neat. We did not
Yeah.
see that same response in the dryland site. But one thing to keep in mind is that water is a big driver of mycorrhizal associations too, because it's all about chemical signals. So when the plants wants to associate with AMF, they're going to release a chemical in the soil. If there are any spores available, they're going to receive that sign. It's like a radio sign, and then they germinate towards the plant and form the connection. If you don't have enough water during that key phase, which for corn is usually between V6 and it tends to peak around R1, but sometimes even before that, that connection is not going to be formed. So water can be critical, but we still need to look at the whole data to see if we can, and we're also looking at precipitation. We're looking at everything on both sides. But what was interesting to see was that we saw many benefits in the irrigated site, but not so much in the dryland site for now. And I'm really curious to see the results after 4 years, because now we have a much more robust answer from that. So this is my main project. We're testing the bioinoculants, but I've done several other side projects too. So one of my favorite ones were with a long-term research site that Dr. Rice has. So Dr. established an experiment over 35 years ago at the Agronomy North Farm, and he was testing the effects of conventional tillage, no-till, and nitrogen fertilization on soil health. So for nitrogen fertilization, he has inorganic fertilizer, so he uses urea, organic fertilizer by using compost, and then control where we don't add anything. And I was measuring AMF on that study, and that was one of the most fun experiences that I had here. It was amazing because it is rare for you to find such an old experiment that you can extract that information from. And I think it's extremely beneficial to farmers because they can get some brand new information that is not out there yet. And what we learned was pretty interesting too. So we learned that no-till was even more significant for keeping AMF colonization than fertilization itself. So when we looked at the amount of AMF in the soil, we saw that fertilization was the main driver on decreasing the amount of AMF in the soil. But when we looked into root colonization, the actual connection between the fungal and the plant, we saw that even though fertilization tended to decline the amount of colonization, in no-till, that value remained high. So keeping that soil undisturbed was much more important for bridging those connections than fertilization itself, which was really cool to see. And I'm also involved in the soil health partnership. So we are looking at the effects of cover crops, like I mentioned earlier, and we saw really positive effects of cover crops on increasing the amount of AMF, even when they had non-host crops in the mix, which came as a surprise for me because some of the farmers were using a cover crop mix with radish in it. And I expected to see, turnip in it, and I expected to see a decline in AMF with turnip, but no, we actually saw an increase because it was a cover crop mix, and all the other crops in the mix were AMF friendly. So it was really cool to learn about that.
Well, I think shifting gears a little bit, I'd like to talk about your teaching experience. You mentioned you taught over 800 students in your last 5 years at K-State, which is amazing. And I think my first question is what classes have you TA'd for? And what have been some of your big takeaways from your teaching experience about the future generation of K-State agronomy grads?
Oh, good. Yeah, I love teaching. This is absolutely my favorite thing to do. I love research too, but having the chance to connect with students, getting them excited about what they're learning for me is it's extremely important. So I've taught a lot of classes at K-State. So I was responsible for teaching soil microbiology lecture and lab since fall of 2021. So it was 5 years teaching that course. Soils lecture and lab with Dr. Merberg as well. I was teaching with him for 3 years. World food crops with Dr. Min, so 4 years. Human and cultural dynamics in the food and agricultural sciences with Dr. Wiley, so Gen Ag. I was also teaching with her for 4 years. And biology of the fungi in the Department of Biology with Dr. Ari Jamponen for 2 years. So I've been around at K State. I really enjoy being in the classroom, and I think it is very important for us to see the university not just as a research hub. Research is extremely important too, but we need to invest in teaching. And I'm really grateful that K State gave me that opportunity to develop methods to bring innovation into the classroom. And I think this is very important because undergrads are the future of agronomy. So we need to get them excited. We need to show them that that works. That work matters. What they are doing is extremely important, regardless of which career path they're going to take, if they're going to go back to their family farm or if they're going to become a faculty member in the future or go to industry. Uh, it is important that they have a good experience, that they get exposed to, a systems view, developing critical thinking. And this is one of the things that I like to bring to my classes. Like, how can I get my students excited to be there? I want them to know that I care about them as individuals. They're not just a number for me, they're not just a tuition for K State. They are real people with real parents waiting for them at home, people that are here chasing their dreams. And it is my goal to make sure that they have the best experience. And I think as ag educators, we have a huge opportunity to make science relevant, exciting, and accessible for all learners.
Would you say that your students are generally receptive to the kind of this focus on particularly your agronomy students or students with a farm background? I guess the reason I ask is one of the things I feel like I hear sometimes is there being a divide between younger farmers and older farmers when it kind of comes to thinking about soil microbiology and soil health and how like those components of farming are driving the system as compared to some of the more traditional things we think about when we think about agriculture. So I'm just curious in your firsthand perspective, what you feel like you hear from maybe some of the future generation of ag leaders and farmers and how they're thinking about it and their interest level on these types of topics.
Yeah, that's a good question too. I think that at least from all the classes from the students that I have experienced, they're very open to new technologies, which is amazing. And especially in our soil microbiology class, which we usually teach for seniors, as compared to the intro to soils, which is like mostly freshmen and sophomore. And most of my students, I had them in both classes, so I could track like how much they changed
Yeah.
over the years, and I could easily see how adaptable they have become. And especially when you show them, and I think that is extremely important for extension, K-State extension too, is when you show them locally adapted data. So data that is coming from Kansas, on farm studies like we're doing with the Kansas Soil Health Partnership, you're showing them that, hey, this work matters. We can actually
Mhm.
build soil health, and this is how we do it. This is how you look at precipitation patterns to answer your questions on fertilizer timing, on bioinoculants. So that class gave me the opportunity to show them what is the reality for Kansas. How can you use that on your own family farm? Like how much of this can you extract? And I think students are really excited about embracing that. And there is a big focus on soil health right now, which is something that really excites me. It's a priority, not just for USDA, but also for students and for farmers. And it is exciting to see that many of my students, they're going back to their farms and they're bringing that new technology to their parents, to their grandparents, and they are adapting. So it's good to see like how many people are now using no-till over the past 20 years, 30 years. It has been growing a lot. And I think educating the newer generation of agronomists is very important for that, because we can make sure that the farmers, old and new, they're having access to that high end information, and we live in an era now, especially with AI and social media, that everything is available all the time, and the attention span of students reduced a lot, so
Mm.
it's a challenge to keep them interested in what we are showing, and to make sure that they know how to filter the information too, because we are in the era of fake news too. So we need to make sure that students are well equipped in finding great papers, finding reliable sources that they can rely on, and bring that information to their farms. But I think they're doing a great job. I haven't faced any resistance on new things, which was common in the past, not on my generation, but I believe Dr. Rice probably experienced a lot of that in the past, because agronomy was focusing a lot on the chemistry, like fertility.
Yeah.
Microbiology used to be forgotten in the corner of the room, and now it's changing. And I think it's fascinating that we're finally giving microbes the attention that they need to, because they're extremely important.
Well, India, I just so grateful for you coming on the podcast and grateful for your last 5 years of service at K-State. I mean, I've gotten to sit in on one of the classes you were TAing for, so I've gotten to kind of see firsthand that impact that you do have on students and you do have on the university and the department. And I think that leads me to my next question, which is kind of the dreaded question for every PhD student finishing up. But I'm sure our audience is just really curious, like what are your plans going forward? What do you hope to do next? Where do you see yourself, 5, 10, 15 years from now?
That's a good question. So, like I mentioned, I'm very passionate about teaching. I'm very passionate about universities. So, my dream is to stay in academia. So, I want to become a professor. I will graduate in December of this year. But yeah, I just want to make sure that I remain in the classroom, which is one of my biggest passions.
So I think the last question I ask all my guests and is just kind of open the floor up to you and just I mean I know we've talked about a lot of exciting things already, so if you just want to reiterate one of those, that's totally okay. But I just like to ask like what's most exciting to you in your work currently or in your broader field that you want our audience to hear about?
Yeah, so like I mentioned in our last question, I think it's really exciting that soil microbiology is getting so much focus right now. It is something that is much needed. And this year, the World Food Prize laureate, Mariangela Hungria from Brazil, she is a soil microbiologist. And I was lucky to be there when she got the award. And I remember she was saying that what we are facing now is called the microgreen revolution. We had the green revolution in the past that changed everything in agriculture. But now we're facing the microgreen revolution where microbes are in the center. They're finally getting that spotlight, exploring the potential of bioinoculants, exploring microbial ecology as a way of intermediate chemical processes, physical processes in the soil. So I think it's really exciting that microbiology finally had the attention that it needed for a long time. And I'm very excited to be a soil microbiologist in this era and be able to contribute to that field. So this is one of the things that have been exciting me a lot is to see that we're finally focusing on soil health. We're finally focusing on not just quick yield responses, short term monetary gains, but on building resilience, on building long term soil health.
I think that's something that excites all of us kind of in this Regen Ag space. I think you put it so well. And Endy, thank you for coming on the podcast. If to the audience, if you're interested in hearing more from Endy or getting to maybe meet her in person even, we're having a field day at Guetterman Family Farms on June 4th that Endy will be one of the speakers at. So that's another great chance to hear from her. And if you're interested in that, there's more information on our social media and our website. And again, everybody, thank you for tuning in to the K-State Regen Ag Podcast, and we will talk to you next time.
Oh, give me a home where the buffalo roam, where the deer and the antelope play, where seldom is heard a discouraging word, and the skies are not cloudy all day. Home, home on the range, where the deer and the antelope play, where seldom is heard a discouraging word, and the skies are not cloudy all day.